My topic today is immune imbalance during aging. We got into that because of our interest in ALS. So a few words on ALS, the fastest progressing adult onset neurodegenerative disease, which primarily targets motor neurons, but also other neurons can be affected as well. There is a significant overlap and with frontotemporal dementia. And overall, there is a large clinical, genetic, and environmental heterogeneity in patients. In genetics, I will just single out immune mediating genes as being found increasingly also in ALS. ALS is marked by neuroinflammation, but not only that, we also see systemic immune imbalance, and we think of it as a potential therapeutic target because it's more plastic than the neurons themselves. So two main hallmarks of ALS are TDP-43 proteinopathy in most of the patients except in FAST and SOD patients, and neuroinflammation at several levels. So we see microgliosis, astrocytosis, oxidative stress, mitochondrial damage, and as Eva showed us, inflammatory hormones are affected as well. And the two major hallmarks crosstalk. It's hard to tell which is the chicken and egg in this dilemma, actually, because one can actually affect the other, and there is a vicious circle between the two. I will also introduce two terms linked to aging, immunosenescence and inflammaging. So immunosenescence is the occurrence of lower output of naive cells, which here meaning adaptive immune T cells and B cells, and increased numbers of effector and memory subsets. At the innate cell level, this is seen as increase of myeloid cell subsets, but their decreased functions and decreased capacity to cope with various stressors and functions that are affected, for example, are phagocytosis and even proliferation. All of this is linked to higher overall inflammatory responses and propensity or skewing towards a pro-inflammatory state at the expense of anti-inflammatory responses. So there is a low-grade chronic inflammation ongoing during aging. We are actually interested in one gene which is called optineurin. It's been found in 2010 in ALS patients, and it's considered a small gene. It's present in a small frequency of patients, but it's in an important pathway because it interacts with tank binding kinase, for example, and P62, which are also genes that have been found mutated in ALS patients. We think that it causes disease by loss of function because it's not found in patients carrying the mutations. It's not found in the aggregates. Rather, it's found in aggregates of sporadic patients, presumably with protective functions. And there are several possible ways that optineurin can affect neuroprotection, because it's an adapter, ubiquitin binding protein that participates in a large number of processes. So the bets are still out. What is the most important? Is its role in inflammatory signaling, autophagy, cell trafficking, or something else? So several optineurin mouse models have been made. The first one published was optineurin deficient model, and there are actually several groups that worked on it, and the phenotype was extremely mild, if any. It's been shown that these mice had problems with vertical rearing of activity and some dysmyelination, but some groups failed to actually replicate that. So we made another model which is more similar to some of the human mutations, and it lacks the C-terminal part of the optineurin, which harbors the ubiquitin binding region, which is we think it's important for function. And like this mutation in human patients, it's expressed also at lower level, likely because of nonsense-mediated mRNA decay. But these mice as well as young mice did not have a phenotype. So this is a model, yes. And what we've shown earlier is that we do have dysregulated cytokine secretion. We had decreased both pro-inflammatory and anti-inflammatory factors distal to the interferon beta pathway. So we tried to look at the phenotype of these mice during aging. So my PhD students analyzed a large number of mice during late 1 year, 2 year periods. So we separated them on males and females, but still we found that aging did not precipitate motor deficiency measured by rotarod in these mice. We also did wire handling tests. There were no differences. We did 2 cognitive tests. One I'm showing here, novel object recognition. We also did not see a major difference. So this stimulus was not enough. Aging with its inflammation and everything was not enough to show the phenotype. We looked more carefully then at the level of microglia, and we found increased, for example, cell body area during, this is I'm showing you between 1 and 2 years, but optineurin mice did not show a difference to wild-type mouse. A similar result was shown at the level of astrocytes. I'm not showing neurons here, but we also did not show differences between neurons in the frontal cortex or at spinal cord. We also tried to see if there is a TDP-43 neuropathology in these mice. So Nicolina actually here in Ljubljana did in Boris Rogel's lab, did fractionation, but we did not see decreased solubility of TDP-43. And when we look at it with fluorescent microscopy, TDP-43 was still largely nuclear, even with, we've tried several antibodies. This is phospho-TDP-43, and also we did not see the aggregates. Perhaps not unsurprising for a loss of function mouse model, but we want to look a little deeper, and we analyzed the cytokine profiles by a protein array, and also aging-induced major differences in cytokine profiles. So we see several pro-inflammatory cytokines coming up. We singled just a few, but most of them were higher. Anti-inflammatory cytokines were higher as well. Some growth factors and chemokines that recruit monocytes to the brain here were also increased in the aging brain, but this optineurin insufficiency still did not affect that. We wanted also to look carefully at the peripheral immune system, because our hypothesis was that the optineurin will actually affect immune cell subsets, not just in the brain. So we looked at by flow cytometry at, here I'm showing just CD8 cells, but the similar story was with CD4 cells. Aging affects numbers of naive cells. There is a huge drop between, now it's a busy slide. First we have a 3-month-old mice. We end by 2 months. 1 year is in between, so we see a drop over time, but equal between wild type and optineurin insufficient mice. And there was an increase of memory cell subsets. We also looked separately for activated cell subsets that express some activation markers like CD38 and CD69. There is their increase. And when we looked at the cytokine secretion profile upon re-stimulation of these cells ex vivo, we also saw much larger production of interferon gamma in aging cells, but unaffected by optineurin insufficiency. So we did not see the differences in the adaptive immune system. We looked in the innate immune system, and the aging affected slightly the number of macrophages. There was an increase during aging and a decrease of conventional dendritic cells. I'm looking here at the peripheral immune system, so we are in the spleen. This is not the brain. We had a technical problem getting the cells out of the brain because they were full of lipofuscin. So this actually clouded the story on the flow cytometer. But okay, we had a drop in dendritic cells. However, when we looked, and this was actually different in optineurin insufficient mice, there was a stronger drop in conventional dendritic cells. And there has been one previous conditional mouse model in dendritic cells that also showed differences in dendritic cells, but mostly in function. And this is the function we looked at just briefly now ex vivo from those old animals. We looked at activation markers like class 2 expression, and this was higher in optineurin deficient animals. In macrophages and in conventional dendritic cells, we had two activation markers coming up, CD86 and MHC class 2. So this is as much as we could get from this in vivo model, but then we took the cells out and looked at phagocytosis of neurons that we apoptotically killed, and looked how microglia was taking them up. We didn't see a difference, but this was a neonatal microglia. And then we looked at macrophages, so bone marrow-derived macrophages, and we've seen a big drop during aging. So on a per-cell basis, phagocytosis of neurons was much lower in aging animals. However, again here, we did not see functional difference in phagocytosis. Phagocytosis was important for us because in sporadic ALS patient, in microglia-like cells, there it's been described that phagocytosis is decreased. LPS, so further inflammatory stimulation, did not affect this. And my final data slide is actually going on those dendritic cells that we saw perturbed in vivo, but we are still looking at just the young animals, and we still don't have phagocytosis of neurons themselves, but we looked at phagocytosis of fluorescent amyloid beta, and we've seen a decrease in percentage of phagocytosing cells in these optineurin insufficient mice, both in unstimulated condition and upon additional pro-inflammatory skewing. And we also saw a difference in the total amount of phagocytosed material. So this is where we are at. Our conclusions for now, for this part, is that aging affects multiple immune functions and does not precipitate neuroinflammation in an optineurin truncation model. It did not trigger neurodegeneration. And these mice showed similar signs even of immunosenescence. Aging decreased phagocytosis, but was not additionally affected by optineurin insufficiency. In the optineurin insufficient mice, we saw decreased dendritic cell numbers, but increased activation. Dendritic cells, still only young ones. We did not look if this is exaggerated in the old ones. We've seen that optineurin insufficient model had lower phagocytic capacity. So I'll, for the sake of time, skip this. Nicolina will talk a little more about our hunting for TDP43 pathology, but my general conclusion would be that the immune system has a major role in the ALS. Just we are still trying to figure out which genes affect it, and if there is a difference. We like to think that it's not just excessive inflammation that starts straight out in these, and which is triggered by dying motor neurons, but given the so many immune-mediating genes that have been found in ALS, we think that there is a possibility that immunodeficiency itself is a trigger of neurodegeneration. But the mouse model is, of course, challenging for any loss of function model. So big thanks for this. This is all work that I've shown here is the work of Nicolina, who will now talk a little more, and Josip Peradinović. Nikolina Mohović and Josip Peradinović. I also thank the collaborators. Some of the work was done in collaboration with Emanuele. Jasna is there always for some other projects, and this one, and Boris also hosted Nikolina for a while. So thanks. Yerneia also, sorry for not putting you here, but definitely many thanks.